Okay, we will continue, Mr. Antonio.
Okay. So the question is, what is a test cross?
I'm sorry, please repeat again. Halo.
Halo, Dr. Antonio.
Okay, Mas Parno.
Able to
Yeah.
ask. Yeah, my name Parno Suarjo. I will to ask to you that about genetic. It's okay.
Yes.
Yeah, actually, I'm proud work with mutation breeding, but the other researcher from by a crossing method said that mutation breeding developed by recessive homozygous. Is correct or not?
I'm sorry, can you repeat the question? Mutation breeding?
Some of researchers from mutation breed from crossing method said that mutation breeding, genetic of mutation breeding developed by recessive homozygous. Correct or not? And then what happened with the recessive homozygous, this genetic? Okay, thank you.
The mutation breeding occurs by the formation of recessive mutants. Is homozygous recessive? Is that what you?
Why the homo
Well,
recessive?
okay. I agree. Most of the cases, when you have a mutation that causes a loss of function, so let's say a protein that stops being workable, functional, that will cause a loss of function, and that usually behaves as a recessive allele. So in that case, the mutation, yes, the mutation breeding is caused by obtaining an allele that doesn't work the same way as the wild type. And somehow, this different function will help to give a phenotype that we like, okay? In some cases, there is a gain of function. But in order to have a gain of function, usually it has to be a gene that has some transacting activity, like a transcription factor. So it will have a different modulation of other genes, and then you're gonna have a new function Or sometimes a gain of function could be a change in the promoter region, then makes that gene respond to a transcription factor that wouldn't just respond before, and that would also be a gain of function. So not always the mutation breeding occurs by the obtention of recessive alleles. But I would say most of the cases, yes, but there are some cases where you have gain of function.
Okay, thank you.
Mr. Antonio.
Okay, nobody wants to answer the question about the test cross. I thought it was easy.
Someone asked in chat box.
Okay. Hello? Okay.
Yes, I'm here.
That's crossing. But yeah, but okay. But you have to have the heterozygous, okay? Crossing the heterozygous with the double recessive, okay? But that's fine.
Okay,
that's
thank you.
fine.
Okay, thank you.
So let's continue. A few more slides and go to the other discussion. Okay. So what I was showing you here is about the change in the allele in the middle, in the gene in the middle, after we have a double crossover, one in each side of the gene. So when we have three genes like this, we can consider recombination is region 1 and region 2, and then the whole segment. Okay? So let's see how we can deal with that. So the genes P, R, and J, they must be arranged in such a way that the center gene switches from the parental arrangement to give classes 7 and 8. So the parental arrangement was like this: P plus, all in coupling, right? P plus, R plus, J plus, or P, R, J, small caps. So we, in the classes 7 and 8, which gene changes from the parental configuration to the other one? So P remains plus, R plus, you have J that changes, and P, R, small caps, you have J plus changing. So the double crossover gametes are then P plus R plus J and P R J plus. Possible gene that are compatible with the data is PJR instead of PRJ, with the genotype of the heterozygous parent being P plus J plus R plus over PJR. So if we now rearrange the genes with this J in the middle, we rearrange the classes here, but according to this. And then you have the parental like progeny, which are these two most frequent classes. And then you have 6 classes of recombination. And now we know that P and J are region 1, and J and R are region 2. So P plus J and P plus J and P J plus show recombination because they are in repulsion here, show recombination in the region 1. So these two numbers, 52 and 46, are the result of a recombination in region 1. J plus R, J, R plus R in repulsion in the region 2. So this shows 22 and 22 recombinants at region 2. And then we have, as we already discovered, we have a double crossover between P and J and R in both situations, where you make J changing from the plus allele to the recessive, and here the recessive to the wild type allele plus. So in order to calculate the recombination frequencies, we need to sum the single crossover in region 1, PJ, plus double crossovers over the total progeny times 100%. That will give us the recombination frequencies in region 1. So in region 1, we have 52 per plus 46. Sorry, that's wrong. 52 here. I made a mistake. 46 plus the double crossover classes, which are 4 and 2, okay, over the total progeny, which is 500, times 100%. So it's 104 over 500, you have 20.8. Recombination frequency between P and J is 20.8%, or 20.8 centimorgans. In the second region, J and R, we do the same thing. We take the recombinants from the second region plus the double crossovers and divide by the total of the progeny times 100%. So we have 22 plus 22 plus the double crossover classes, and then over 500. So that's 50 over 500 times 100%, that's 10%. So the recombination frequency between J and R is 10%, or 10 mapping units, or 10 centimorgans. And the total recombination in the region, considering the two extreme genes, is the sum of everything: 52, 46, 22, 22, and 2 times 4 plus 2. Okay, so the 154 over 100 over 500 times 100% is 30.8%, or 30.8 map units. Okay, so the question is, is the probability, or if the occurrence of one crossover, does it interfere with another crossover nearby? Do you have some type of inhibition, or there's never an interference between one crossing over and the other? So it means, it looks like there is some interference. So when one crossover occurs, it will prevent or lower the chances of having another crossover nearby, because it makes sense. It makes sense in such a way that there's a lot of energy spent by the cell to make that crossover, and that crossover will be advantageous for the progeny, because it will generate variability But if you have a second crossover that turns back the result into what it was before, it is 2 times the job and no result. So that's a waste of energy. So the cell will have mechanisms that prevent 2 crossing overs to occur very close to each other, because one will undo the job of the other one. Okay. So the recombination frequencies are not identical to crossover frequencies, and typically they underestimate the true map distance. Okay, so that's the first point we need to make. We detect, as I told you before, we detect recombination frequency, but the crossover frequency can be there and we do not detect it. So there is always an underestimation of the crossover frequency and therefore the true map distance. Okay, we have like a confidence interval where the recombination frequencies are closer to the values of true crossover frequency? Yes. The usually small regions below 10, below 7% recombination, you have exactly the same values for percentage recombination and percentage crossover. So in this case, in our example, we computed recombinations in regions 1 and 2. And if crossing over in region 1 was independent from crossing over in region 2, then the probability of a double crossover in the 2 regions is equal to the product of the probabilities of the 2 events occurring separately. So that's a good check we can do. Since we know, we were able to calculate the recombination between P and J, right? That's our order is P, J, and R, okay? So region 1 is P and J. Recombinate crossing over in region 1. That means between P and J is independent from crossing over in region 2, which means between J and R. Then the probability of a double crossover will be the product of both probabilities. Okay, let's see if that's true. So the recombination frequency in region 1 times the recombination frequency in region 2 equals to 0.208 times 0.1. That's 20.8% or 10% recombination, respectively, we just calculated. So 0.208 times 0.1 is 0.0208, what would be equal to 2.08% of double crossover being expected to occur. Okay, how much did we get with this data? We only got 6. The 2 classes were 4 and 2. So back, let's go back. This 4 and 2 are the only ones with double crossovers. So it's 6, 4 plus 2 is 6, over the total 500. So 6 over 500 is 1.2% double crossovers were observed, classes 7 and 8. So if that was true, if the complete independence between the crossing overs occurring in region 1 and region 2 were true, then we would have the same value, but we have a lower observed ratio of double crossovers than it was expected. So that means that there has to be some interference between the occurrence of one crossing over and the other. So that would make the frequency of double crossovers to be even lower than the expected rate calculated by the product of both probabilities. Okay? So we can call it as a coefficient of coincidence. So the coefficient of coincidence is the observed double crossover frequency over the expected double of crossover frequency. And interference is 1 minus that. So our coefficient of coincidence is 0.012, which is the observed double crossing over rate, over 0.0208, which was the expected double crossover rate. And we got a 0.577 value for the coefficient of coincidence. 1 minus that is 0.423. So our interference is 0.423. In practical means, it means that only 57.7% of expected double crossovers really occurred. Okay? So luckily for us, when we estimate map distances, the double crossovers that are not detected by our experiments when we're having two genes, we're comparing two genes, we cannot detect the double crossovers between the two genes. So when this happened, not only the double crossover is low, very low, and the closer the genes are, the lower it becomes, because if you have 20% distance between two genes, you have 0.2, and the double crossover rate would be 0.2 times 0.2 is 0.04. If you have 0.1 distance for 10% recombination, the frequency of double crossovers will be 0.1 times 0.1 will be 0.01. It means 4 times less probable than when you have a 20% recombination or 20 centimorgan distance. So it means that not only the expected frequency of double crossovers is small, but also the interference will make it less even or less likely to occur these double crossovers. So in this case, in our example, only 57% of the double crossovers that were expected really occurred. So in order to calculate accurate map distances, we have to make corrections for this interference, since it is not exactly when we not exactly linear, the relationship between the frequency of recombination and the frequency of crossing over. So the map units between linked genes usually are a measure of crossover frequency. And the geneticists, they quantify, what they quantify is the frequency of recombinants. What we've been looking at here is the difference of recombinants in the progeny. So are they always identical? The crossover frequency and the recombination frequency, are they always identical? No, they differ from each other. But when you have larger distances. When you have small distance, they are practically the same. So how can we obtain maps? With maps, we have different distances. We have not only small difference, but we have whole genome distances. So if we look at the progeny of single and double crossovers, if you look at one single crossover, you find or you detect parental and recombinants, like here, big A, big B, small a, small b, parental genotypes, and the recombinants, big A, small b, small a, big B. If you have double crossovers, then here you would see all parental. Okay, so you wouldn't detect. So if no more than a single crossover occurs between linked genes, there is a direct linear relationship between the genetic map and the observed recombination frequency. So if you don't have double crossovers, you don't have a deviation from a linear relationship between recombination frequency and crossing over frequency. Only when the net genetic map distance are small, for example, around 7 map units, or some people consider less than 10 map units, then you have equivalence between recombination frequency and crossover sequence or map units. So for most mapping projects, you can have on your software, there are usually possibilities of considering two types of functions. You have the one proposed by Haldane, which assumes no interference, and one proposed by Kosambi, which assumes interference. So if you're doing a mapping project and your software asks, okay, use Haldane or use Kosambi. So you have to know that when you assume there is no interference, it means that one crossover does not interfere with the occurrence of a crossover nearby. If you choose to use Kosambi function, then you consider some interference. So if you compare here, Kosambi and Haldane functions, you see here from up to 0.1, all three are the same. So the mapping units and recombination frequency, detected recombination frequency, they all work the same. Above 0.1 recombination fraction or 10% recombination, then you start having a deviation from the mapping units. So D, mapping function, here's D. The D is the crossover rate, and R is the recombination fraction. So D is the crossover rate, crossover frequency, or the unit, mapping unit, or the centimorgans. So it's D. And R is the recombination fraction or recombination frequency. So if you go up to 0.1, they measure the same. But then when you move above 0.1 recombination frequency, you have a change with Kosambi and a change with Haldane. So you get a larger map units from Haldane because you're not considering interference. So you have a little bit of overestimation of Haldane when compared to Kosambi, because this one, Kosambi considers interference, so it has some correction for the, it will be accounted, less double crossovers will be accounted for in the function of Kosambi, and more double crossovers will be accounted for all the crossovers, right? There's the expected double crossover frequency will be incorporated in the function of Haldane. So let's say here you have a 0.2 recombination fraction, so it's 20%. The double crossover rate estimated in Haldane will be 0.2 times 0.2, so it'd be 0.04. So the distance estimated by Haldane will be 20 centimorgans plus 4 centimorgans. So it'll be 0.2 plus 0.04. So it'll be 24 centimorgans, 20% recombination. If you consider Kosambi, it will be somewhere between 20 centimorgans and 24, because then you will have a correction for the interference. So the number of crossovers estimated will be lower than the expected one. It will be closer maybe to the observed, but would be a lower estimate. So that's why you have an overestimation. So what can we take, conclusion we can take is, if we are doing a mapping and we use Haldane, we expect to generate larger maps, because for every distance we'll have every recombination fraction we calculate, we'll have a larger map distance in our map. Okay, and if we switch to Kosambi, our map is going to shrink. It's going to be smaller because you're considering interference here. Is it clear? Anybody has this or want me to explain it again? This is very important for the follow-up lectures that we'll have. Okay.
Okay. So
Okay,
So,
may I ask a question about?
Yes. Yes.
Okay, so so in this case, when we should use Kosambi and when we should use Haldane, because in the software sometimes it is as easy as to just switch the option. So is it always better to use Kosambi because it's quite safer than Haldane, or is it depends on which kind of situation we should apply in our mapping project?
Yes, good question. Well, it really it gives you a small difference. It's not such a large difference, but this is quite a difference. You only have to know or to be aware that when you use Haldane, your estimation will be a little bit bigger. Okay, so it means that you will expect a recombination frequency, because it goes both ways, right? You take a small experiment, you define the distance between your genes, okay? And then you expect, when you're working with your big breeding population, you expect to find a given number of recombinants based on the genetic distance between the genes. So if you estimate, like I told you, estimate with a 0.2 or a 20% recombination fraction, you estimate with Haldane 24 centimorgans, 24 map units. It's you go back, and then next population you're going to work, you say, okay, my genes are 24 centimorgans apart. So, I can expect to look at a population of 1,000 individuals. I can expect 24% recombinants. So I can expect 240 recombinants in 1,000. If you use Kosambi, you expect maybe something around 200 and 220. Okay. In this case, practical terms will give some slight difference. It's not wrong, right or wrong, to use one or the other. It's just that you have to know you're getting a higher estimate with one of mapping distance with the other. But for practical terms, they are very close. Okay. Of course, if you map a, let's say, make a map of recombinant inbreds or F2 population, and you use one function or the other, then your map will change slightly, but it will change. And what is the major tendency you're going to find? The major tendency is that the Haldane map will be bigger because you are increasing the map distance between the genes. In practical terms, you have approximate values. But both are models, right? When you have models, you are trying to explain biological variations with mathematical models. And that's always a challenge because biology has a lot of factors affecting, like environment, like other biological, internal biological factors that will change the expected from a simple mathematical point of view. Okay?
Thank you.
Any other question? So this implications for plant breeding, this is the last slide. So you have positive associations that are good. In between favorable genes that can be easily maintained. So if you have two good genes that are linked, it's nice because you know you can select one and have the other coming around. On the other hand, when you have unfavorable associations, that's tricky. You have one favorable gene and a linkage drag or some gene that comes along that's not so good, it's not desirable, then you have to break this association between these genes. You want to have the good alleles for both loci. So in order to do that, you have to do crosses and use large populations to achieve this goal. Because the more crosses you do, let's say, when you have a homozygous, like a self-crossing species, soybean, rice, wheat, okay? You have a homozygous genotype and the genes are linked, you're not gonna break that, that linkage because you have all the chromatids the same. So you need to cross this, this genotype to a recessive with different alleles, and then do a test cross again, be able to break the association between the good gene and the bad gene. Only with heterozygous in both loci, then you can have the change in, let's say it was a repulsion phase, you can change from a repulsion to a coupling and then have the good alleles in both genes. So that is obtained with meiosis, and meiosis is obtained with the sexual reproduction of heterozygous individuals. So either crossing it to recessive or allowing the heterozygous to produce progenies, you can have the recombination between these two genes. And of course, if these two are tightly linked, it means the recombination frequency is very small, let's say 0.1%. So 0.1% recombination, it means that you only get one recombinant in 1,000 plants. So if you have 1% recombinant, you have one plant in 100 progeny. If you have a 0.1, so your two genes are linked are by 0.1 centimorgan, one recombinant at 1,000 plants. So in order to be sure, you need a population of at least 5,000 to 10,000 plants to have 5 or 10 recombinants to play with. If you produce a progeny with 100 individuals and your two genes are 0.1 centimorgan apart, you're not going to break this bond. You end up with all the progeny with these genes linked still, the favorable and unfavorable genes in the same individuals. So that doesn't help you. So you need large populations to be able to have the probability of recombination between the two genes and then have them apart. Same thing will be with the mutant. Let's say you get a mutant and there were two mutations nearby and you have one that's bad, and you want to take one gene that's good and you separate from that other mutated gene that is bad. So we're going to have to do crosses and use large populations to separate these mutations, this good and bad mutation. Okay, so let's I separated one paper for, so there's not today, still I looked and there's another round table, not for the marker seed selection yet, because before we're going to have the class for marker seed selection, then we can discuss the paper. But this paper, I I chose, it was an overall, let's see, an overall overview of genomic resources and plant breeding for sustainable agriculture. So this is a paper on was plant physiology, journal plant physiology, and there's a very good group of scientists here. James Schnabel, you have Vikas Singh, you have Henry Nguyen, you have Shobana Sivasankar, you have Scott Jackson, you have Timothy Close, you have Rajiv Varshney. So that's a good team of scientists. These are the ones I know, but there are some other good ones. And so they put together a good idea of what will be the challenges and what will be the major tools to go forward from now on. I'm going to present a few slides for this paper, and then we can discuss. Okay? Let me open it and share. I was not sharing it. Okay. Sorry. Okay.
Okay.
Do you see now?
Yes. Sir.
Okay. Yeah. So these are the authors of the paper. I'm sorry, I was showing the screen and not sharing it. So the title of this paper is Genomic Resources in Plant Breeding for Sustainable Agriculture. It's one of the papers I handed you. And one background, they bring us about 200 years of plant breeding. It's interesting. Well, they dig up this reports by Darwin working on the Anthus, the carnation, and showing the sum of in crosses and the progenies. So the ratios of the progeny was obtained. And together with this work from Darwin, there was this rediscovery of Mendel laws. So, and then there was this, let me put this in. Okay. And the Green Revolution that created a high increase in crop yield, around 56% between 1965 and 1985. But now we have global climate extremes, which translate in drought, flooding, extreme temperatures, and intensified incidence of pests and diseases for the last 40 years. That has had a serious impact on agriculture. But despite all this progress that we made in yields increase of yields, in 2019, there's a report of 144 million children under 5 with stunted growth, and about 340 million suffering from micronutrient deficiency. So we still have a long way to feed all the people in the planet, and in a satisfactory way. One of the sustainable goals by FAO, by the United Nations, is zero hunger, and despite we had and we keep having increases in the major cereals such as maize, rice, wheat, and soybean, and major cereals and legumes, with growth rates of 1.6, 1.9, 1.3, average would be around 1.2. They are nearing a plateau. So it means that we are the same improvements we obtained in rice in the 80s was cut by half in the 90s and almost by half in the 2000s. So it's getting more and more difficult to increase yields. So it means that the genotypes that we have available are reaching the plateau of their yield possibilities, their yield. So how can we break that? How can we change the plateau of a given species? We need to bring novel alleles. We need to bring variability to increase this plateau. And there are two major ways of doing that. One is by bringing genes from wild relatives. And the second one is generating mutants, generating novel alleles in the germplasm we already have. So the group called for a new effort, a coordinated effort in the new agronomic methods, advances in breeding technologies, development of novel genetic diversity, I just told you, and the use of genomic resources to discover and integrate novel genes and alleles. So that's the major point made by the group, is that we are okay, we have improvements, okay, breeding has brought us here, but now what happens is that we have it looks like we're not getting the same kind of improvement we used to have in the 80s and 90s. So it means that we are exhausting the variability we have at our disposal. So how can we overcome that? Because, you know, there is always a discussion between breeders, among breeders, that breeders want some type of elite line, okay? Because there is already a need for a type of plant that the farmer likes, the consumer likes, and so there are some adaptation features that cannot be changed. So many breeders, what they do is they cross elite by elite lines and try to get transgressive segregants from these elite lines. But since these elite lines are now narrower and narrower, with narrow genetic variability, the cross between them doesn't get us too far. So we need to be able to introgress bring novel alleles or to mutate the ones we have to obtain novel alleles. And that's the point. Okay, we need to be able to molecularly understand what the changes are and how they can be, we make profit out of these changes. So the other point that I make is that the 21st century has had transformative technological development and declining costs in genomics. So we've seen, for example, the cost of the rice genome, and it was finished in 2005, was estimated in $100 million. And now we can make a very good genome with $50,000. So that's 200 times cheaper than it used to be. That's the way the technology is advancing that is helping us to decrease the costs in genomics. Also, since the cost of genomics is decreasing, more and more genomes are being sequenced. And we have around 800 genomes available now, already sequenced. Also, we are improving our way of predicting and tailoring climate resilience of crops. So we are getting better in phenotyping our plants. For example, last month I bought a drone for our lab. My students insisted on it to take pictures of our plants by air with a drone, and that can help better phenotyping, and it will help the breeding process. Also, these advances in next generation sequencing helped us decode many genomes and help use markers associated to trait of interest. So the sampling of reference genomes and germplasm sequencing is one of the great or the major advances that are going on right now. Because in the late 90s or early 2000, we thought that, okay, we have, if we have a reference genome, we will be able to make improvements based on that reference genome. So rice would be a reference genome for rice and also for other grasses, wheat, maize. And then we discovered that, okay, no, it's not like that, because different species have, or even individuals have different kind of genome organization, different ploid levels, and variety of repeat contents. So even though we are already adding up 800 species with draft genomes, we are going for de novo assemble of relatives. Another concept that also came in the last decade was the concept of pangenomes. What is a pangenome? A pangenome aims to capture the total diversity within a species. But that can be divided into core genes and dispensable genes. So what do you mean by core genes? It means that if you sequence 3,000 rice genotypes, the core genes will be present in every single one of them. But around maybe 5 to 10% of the genes will be present in some genotypes and in others. And these are called the dispensable genes. Okay. Even though they are not essential, they can be very important for agriculture. For example, one of the hypotheses now is that this set of dispensable genes is the reason why we have heterosis when we combine different genotypes. There are very good combining ability. The combining ability would be related to the complementarity of these dispensable genes. They are present in one line and not present in another, and vice versa. So that would be a good fitting to the heterosis hypothesis that result from the complementarity of two genotypes, crossing two genotypes. But also helps us to understand, okay, sequencing Nipombare rice doesn't solve all the problems of rice breeding. It, of course, it helped a lot to improve and find genes, but it will not solve all the problems of rice breeding. So that's why the projects like 1,000 genomes and then 3,000 genomes, and our collaboration with the IOMAP, so that we sequence over 12 genomes of IAEA genomes that help to provide some information on the wild relatives and genes present there. So here they put a very nice figure showing the sequence-based trait mapping approach. So now we are having different types of techniques that help us to do a better characterization of these genomes. So GBS is genotype by sequence. It's a robust, simple, versatile, cost-effective procedure for SNP discovery and mapping. Whole genome sequencing, whole plant and animal genome sequencing provides most comprehensive characterization of a genome. You have RAD-seq, where you reduce complexity for sequencing. Also can improve the information coming. BSA sequencing, which is a different kinds of BSA was a technique. It was described by Mitchell Moore's group from Davis, UC Davis, in the early 90s, I think it was 1991, if I'm not mistaken. And it's a brilliant technique. And every breeder and geneticist should know about this. So that's bulk segregant analysis. So when you compare, you want to identify markers for a given region in the genome associated with the trait you're looking for. You take a segregant population and divide it in bulks that are extreme for the trait you're looking for. So when you compare the bulk that has, let's say, a disease susceptibility with a bulk that has disease resistance in a progeny of a biparental population. So it's a two genotype cross, and then you have F1, F2, and open F2 go. You can go up to F6 or even an F2. You can do it. So you have two bulks, and then you compare the bulks with different phenotypes.